me a great pleasure to introduce Danielle Edwards. For those of you who know her, you know she goes by Dan, which might be confusing because not too many women go by Dan.
It's very common in China.
Is it common? Ah,
common
yeah, of course.
female name in China.
Great. So great pleasure to introduce Dan. I've known her for many years, more than 10 years. I think I first met you even in I didn't meet you in Australia. I met you when you visited here the first time, I think, and then I saw you in Australia as well. And so she does a lot of work with reptiles and amphibians, as we'll see today. Just a little bit of history. She did her undergraduate degree at the University of Newcastle, which I just learned is in Australia and not in England. Right? And then she did her PhD with Dale Roberts at the University of Western Australia. And she received her PhD in 2008. But I think that was a little bit late after the start because there were some intervening postdocs and informal postdocs that were sandwiched in between starting your PhD and finishing your PhD, which is interesting. So she spent
also a health crisis.
And a health crisis, and a year where you were a free agent.
That was after my first postdoc.
Okay. She has a long history. But she worked with Jane Melville at Museum Victoria. Many of you know Jane. And after she worked with Jane, she worked with Scott Keogh. Many of you will know Scott Keogh at Australian National University. And then she went to Michigan and she was a postdoc with Lacey Knowles. And then after she left Lacey Knowles' lab, she went to Yale and she was a postdoc there with Gisela Chacon, which I think was what brings in the tortoise work, because prior to that, her work was on frogs in the Australian arid zone, and also working with the gametes in the Australian arid zone, and now she has a tortoise component to her work as well. And so I'm happy, and in 2015, she started her faculty position at the University of California at Merced. So she's a neighbor, and we hope to see more of her now that she's in the area. And today she's going to be speaking about ecological divergence, adaptive radiation, and the evolution of social signaling traits. Thanks for being here, Dan.
Well, thank you very much for having me, Jim. It's my pleasure to come here and talk to you about some of the stuff that I've been doing for a long time and some also very new stuff that I was doing in my postdoc. So I have had a long trajectory, as Jim mentioned, but in general my research is interested in what drives speciation. Traditionally I had come at this from a biogeographic perspective, and a lot of my research had been done in this very spectacular southwestern Australian system. But some of the things that came out of that biogeographic work really have shifted the way that I think about science and the way that I do research. Basically, firstly, that speciation is a continuum. You can get stages in speciation from the initiation of speciation through to the completion, and that corresponds with certain aspects of reproductive isolation, from being absent through to complete, and is associated with certain genetic signals that we see in data. Secondly, that not all divergence results in speciation. And some work from Jeroen Rosenblum here, the ephemeral species concept, I think encapsulates that very cleanly. That you can get divergence in populations that doesn't necessarily result in species. You can get extinction of those populations, emerging of those populations through hybridization. And so my research now is very interested in what drives those processes from the inception of speciation through to the completion. And that is basically what I'm going to talk about today. So in general, when I'm talking about population, divergence and speciation, there are a number of key processes that can be involved in generating that signal of population, divergence and speciation. You can have environmental processes. A great example of environmental processes driving radiation of organisms is the anomalous radiation, where you've had species that have vertically diversified in their structural niche, occupying ground, trunk and canopy habitats, and systematically diversified across a number of island systems. You may also have environmental processes that drive speciation through vicariance biogeography, whereby you get species that's contiguous, an environmental process like a river. My blue blob is supposed to be a river here, that might split populations which then diverge in allopatry according to drift, and possibly other processes. You can also have inter and intraspecific interactions that drive speciation and diversification. Intraspecific variation, like these Gouldian finches here, you might get selection on specific morphs that might be more favourable than others. You also can get between species interactions like the bird of paradise here, which involved dramatically different sexual signals that are selected among different species. But something that I've become interested in, and what many people are also interested in these days, the fact that you can get this interaction between environmental processes and the processes, what we might call sexual selection processes, that interact in meaningful ways to drive speciation from its inception through to completion. And it kind of sets the stage for what I'm going to talk about here. So traditionally, when we think about ecological divergence and how it results in speciation, we think about some sort of meaningful environmental change that creates an ecological opportunity, a new open environment perhaps that's created by the generation of an island system, or perhaps a group that invades a new habitat that's very rich and they're able to diversify sympatrically within those habitats, that then results in some divergence. So we've got two very prominent examples here of what are traditionally termed ecological radiations. We've got the finches here, which have radiated among the Galapagos Islands. Their beak morphologies have adapted to specific diet niches that have been available on different islands, and that's putatively driven their adaptive radiation. We also have the Caribbean anolis lizards, which have again diversified among different islands, and then within islands diversified among different strata within those islands repeatedly. But what about ecological divergence and social signals? Well, while those two systems are traditionally thought of as purely ecological adaptive radiations, some other research has actually shown that those have implications for the signals that they use to differentiate among species or mates. So for instance, the Galapagos finches, the beaks actually have meaningful impacts on the song signals that they use in their mating interactions with one another. So that, in terms of their ecological adaptation, has an unintended consequence for the interactions that they have among other species. Furthermore, anolis lizards, we see a dramatic variation in the dewlaps that they have in terms of the signals. Now, this paper is specifically on the evolution of female traits, and what this paper suggests is that social selection and environmental selection work together to drive the evolution of female traits, but the same has been shown for the males as well. So the kind of focus of my research today, as you could gather from the talk, is about ecological divergence, adaptive radiation, and the diversification of social signaling traits. And the specific questions that I'm interested in looking at under this umbrella are: how do specific ecological factors drive adaptive radiation or adaptive evolution? Does ecological divergence contribute to social signal evolution? And does geographic context impact the importance of adaptive and social signal evolution? I'm going to talk about two specific systems that I've been working on. I'm going to talk about some of my postdoc work that I've been doing on Galapagos tortoises. This is extremely preliminary work, and so it's going to represent a much smaller part of my talk. And it will also only be looking at this first question about what specific ecological factors has driven diversification. I've included a wild speculation slide in there about what social things might be going on with the tortoises, but again, caveat, that's my own wild sort of speculation. The second part of my talk, I'm going to focus on some work I've been doing in irradiation of gametes from Australia, which touches on all three of these aspects. So for the first part, I'm going to talk about tortoises. So in case you didn't know about Galapagos tortoises, I thought I'd give you a brief introduction. Galapagos tortoises are a series of species that are distributed across the islands. Several of the larger islands, like Isabella here, have multiple species. Some smaller ones have had multiple species as well. But what we have is the evolution of two sort of phenotypes, if you will. The first is the domed phenotype here. These individuals are larger in general than the second phenotype, the saddled. And they can also show variation in shape, and the saddles in fact do, but there is a very clear distinction between the saddled and the dome phenotype. There is sort of continuous variation within each of those that I'm not going to talk about, but for the purposes of the talk I'm going to talk about today is we've binned those very discreetly into two types. You've got the saddles and the domes. So one of the current theories is that those two phenotypes have evolved in response to rainfall regimes on different islands. And I want you to remember that. This is a hypothesis from Fritz, who based this on his studies of morphology in Galapagos tortoises. Essentially what you tend to see are domes on younger islands and saddles on older islands. And that will come in handy for later. A significant challenge of this work has been the level of extinction that we see in Galapagos tortoises. And it's meant that I've had to incorporate some ancient DNA techniques that has been extremely challenging and not very rewarding. I don't have the data to talk about those today, but this has been a lengthy process, and this is a complicating factor. And many of you will know, I believe I had a conversation with Ian Wang this morning, this is Lonesome George as representative of our saddlebacks here. He passed away in 2012, and the Pinta species went extinct when he did. So if ecological divergence has driven phenotypic evolution in Galapagos tortoises, we might expect that there have been multiple transitions between phenotypes, that phenotypes occupy distinct ecological niches, and that phenotypic evolution corresponds with the ecological niche. This work is something that I'm currently preparing for publication. So in terms of answering whether or not there have been multiple transitions in carapace shape, what I did was construct a species tree. From now on, what I'm going to do is put specific methodological details down here. I may or may not talk about them. If you're interested in exactly what I did, I will talk with you at some later stage about that. But just bear in mind, these are going to be the relevant details down here. So this is a species tree constructed from about 25 different sequence loci for all of the what we call modern samples that we had for DNA from tortoises. These represent samples that we actually had blood samples from, and include the Pinta species because we had a sample from Lonesome George. What I did was I coded those as either saddled or domed, and they're going to be, the saddles will always be green, the domes will always be black. And I did an ancestral state reconstruction. What we know a priori is that the mainland ancestor of Galapagos tortoises is a domed tortoise. It's not a giant tortoise, but it has a domed shell. And our analysis here is, it's not very powerful because of the sample size. Hopefully that will improve when we have some other extinct lineages in there. But this seems to be supported in our analysis that the ancestor of the Galapagos tortoises was indeed a domed animal. And there are multiple transitions between domed and saddled species. And this is even more clear when you consider that according to our extinct species and using the single mitochondrial marker that we currently have, several saddled species are more closely related to domes than they are to saddled species. This is an individual from the species from Santa Fe. We only know these species from bones that were collected from a fire on a beach, and we don't currently know what it was. We think it was a saddle, and I'll explain to you why later. But we definitely do see multiple transitions. This is the new species that was just described on Faustoi. So there have indeed been multiple transitions in carapace shape in Galapagos tortoises, but have their niches diverged? So to do this, what I did was look at the ecological niche and a bunch of traits and use an ancestral state reconstruction of ecological niche. Again, we have the domes in green, the domes in black, the saddles in green. This is like an average for a particular environmental variable. In this case, it's vegetation, NDVI, which is an estimation of vegetative biomass. And what we see is in a phylogenetic context, our saddles are occupying much lower vegetative biomasses than our domes. And when we test this, so the dotted line here is a 95% confidence interval around sampling of that variable across the distribution of each species. And when we test this in a phylogenetic ANOVA, what we see is that the phenotypes occupy significantly different aspects of vegetative biomass and elevation. And note we tested a whole bunch of other variables, rainfall included based on Fritz's original hypothesis. They did not occupy significantly different rainfall niches. What came to the top was both vegetative biomass and elevation. So next question we wanted to ask was, is this the evolution of these different phenotypes correlated with this ecological divergence? And so to do that, I used a MCMC Bayesian approach to test this using a model developed by Liam Revell. What you see here is a sampling frequency from the output of that. We have sampled correlation coefficients along here and the sampling frequency. We have our average correlation coefficient here and the 95% confidence intervals around that correlation coefficient. And what this tells us is that because these 95% confidence intervals do not cross zero, there is a significant correlation between the evolution of phenotype in Galapagos tortoises and the niche that they occupy, particularly in vegetation biomass. And now this didn't come out in any other variable, even elevation, and I'll discuss why I think in a minute, but essentially what this is telling us is that the dome species are only able to be maintained on high vegetative biomass islands, and that when species independently colonize islands that are older and have lower vegetative biomass, they are forced to evolve the saddles to increase the browsing capabilities that they have in these low resource environments, which is pretty neat and cool. I don't think I've ever worked on a result that this neat before ever. So essentially what I think is happening is these older islands are lower, they are more eroded. The higher islands, the younger islands have higher volcanoes. Those volcanoes push up into the mists that come across. The Galapagos are essentially an arid suite of islands in a tropics zone. Those higher elevation islands, the mists collect and the higher vegetative biomass is able to exist on those high elevations. So there's some sort of interaction, and it's not purely rainfall, but that moisture is involved. And the key variable here is vegetative biomass, and you need a higher vegetative biomass to support a larger dome tortoise than you do for a smaller saddle species. So in terms of speciation of Galapagos tortoises, what we see is ecological divergence is driving phenotypic evolution at a staggering rate. I say staggering because the Galapagos, the origin of Galapagos tortoises in the archipelago is putatively around less than 2 million years ago. When you consider that the generation time of a tortoise is at minimum 20 years, that is a staggering rate of evolution in quite an extreme way. They don't breed very often, and they don't recruit a lot of offspring. They're lucky to get maybe one through every four breeding events. So, and so some speculation: are they reproductively isolated, and might species show mating preferences? We have some anecdotal evidence for this. Basically, no, they're not reproductively isolated. When humans have introduced them to divergent lineages to one place through transport, we see that hybrid swarms form very quickly. But having said that, we do actually see some mating preferences when you have colonizations of species